Pollination, I would like to explain you one simple concept in self-pollination itself. Okay, so as we know, what is self-pollination? So there will be a pollination within the same flower. Okay, so the transfer takes place. Transfer of pollen grains from anther to the stigma takes place in the same flower, and it doesn't show any variation, that is genetic variations. So hence, it makes the self-pollinated plants like a pure breed. It is also called as pure breed. But what happens during generation to the generations? Like example, if first generation is produced by self-pollination, just imagine this is the first progeny generation. Okay, so what happens if the same progenies again perform the self-pollination? Second generation, third generation, fourth generation, fifth, sixth. So like this, successive generations are forming by performing the self-pollination itself. But what happens, the problem with the self-pollination is, if they continue one generation to the another generation with the same pollination, so what happens, the qualities which are present in the first or the second generation cannot be inherited by the next few generation. They may lose the important characters, or we call it as traits. Okay, so what happens, ಒಂದು generation ಇಂದ ಇನ್ನೊಂದು generationಗೆ starting ಎರಡು successive generation ಇರಬಹುದು, they are showing the true characters. But ಹೋಗ್ತಾ ಹೋಗ್ತಾ ಏನಾಗುತ್ತೆ, generations ಮೇಲೆ generations, when they perform continuously self-pollination, so because of there is no variations ಇಲ್ಲದೆ ಇರೋ ಕಾರಣಕ್ಕಾಗಿ, so ಏನಾಗುತ್ತೆ, ಇರೋವಂಥ ಒಂದು ಒಳ್ಳೆ ಕ್ವಾಲಿಟಿ ಕೂಡ they may lose. Okay, the successive generation. So that losing the important characters to the successive generation, this cause one condition in the plants. Okay, that we call it as inbreeding depression. ಓಕೆ, ಸೊ ಹೇಗೆ ನಾವು ನಮ್ಮಲ್ಲೂ ಕೂಡ ಹೇಗೆ ಡಿಪ್ರೆಷನ್ಸ್ ಅನ್ನೋದಿರುತ್ತೆ. ಓಕೆ, ವೆನ್ ದರ್ ಇಸ್ ನೋ ಚೇಂಜ್ ಇನ್ ಲೈಫ್ ವಿ ಫೀಲ್ ಡಿಪ್ರೆಸ್ಡ್. ಇನ್ ದ ಸೇಮ್ ವೇ, ಇವನ್ ದ ಪ್ಲಾಂಟ್ಸ್ ಆಲ್ಸೋ ಆ್ಯಂಡ್ ಇವನ್ ಎನಿಮಲ್ಸ್ ಆಲ್ಸೋ, ಇಫ್ ದ ಬ್ರೀಡಿಂಗ್, ದಟ್ ಈಸ್ ಇನ್ಬ್ರೀಡಿಂಗ್ ಅಂತ ಹೇಳ್ತೀವಿ, ಸೆಲ್ಫ್ ಬ್ರೀಡಿಂಗ್, if there is no variations, then what happens, they don't show any variations. ಇರೋ ಒಂದು ಒರಿಜಿನಲ್ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಕೂಡ ಏನಾಗುತ್ತೆ, they will lose. So this, what we call it as inbreeding depression. To avoid this inbreeding depression, some plants are adopted, like they actually want to perform the self-pollination. Because of this inbreeding depression, what happens, they avoid self-pollination and they encourage cross-pollination sometimes. ಏನು ಮಾಡುತ್ತೆ, ಈ ಒಂದು inbreeding depression ಅನ್ನು ಓವರ್ಕಮ್ ಮಾಡೋದಕ್ಕೆ ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನನ್ನು ಅವಾಯ್ಡ್ ಮಾಡುತ್ತೆ ಕೆಲವೊಂದು ಪ್ಲಾಂಟ್ಸ್, ಆ್ಯಂಡ್ ಏನು ಮಾಡುತ್ತೆ, ಕ್ರಾಸ್ ಪಾಲಿನೇಷನನ್ನು ಪರ್ಫಾರ್ಮ್ ಮಾಡೋದಕ್ಕೆ ಎನ್ಕರೇಜ್ ಮಾಡುತ್ತೆ. ಓಕೆ, ಕೆಲವೊಂದು ಡಿವೈಸ್ಗಳ ಮೂಲಕ ಏನಾಗುವಂಥದ್ದು, ಎನ್ಕರೇಜ್ ಮಾಡುವಂಥದ್ದು ಈ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನನ್ನು. ಸಚ್ ಡಿವೈಸಸ್ ವಾಟ್ ವಿ ಕಾಲ್, ಸೊ ಸಚ್ ಡಿವೈಸಸ್ ವಿ ಕಾಲ್ ಇಟ್ ಆಸ್ outbreeding devices. Okay, so these are the devices which avoid the self-pollination and encourage the cross-pollination. So what they do, avoids the self-pollination, yes, and encourage cross pollination. To overcome what? To overcome which condition? To overcome inbreeding depressions. Okay, to avoid the inbreeding depressions or to overcome the inbreeding depressions, what they do, basically they self-pollinate. They are the one self-pollinators. But what happens, to avoid, to overcome the inbreeding depression, they switch to cross-pollination. They undergo cross-pollination and they can make the variations in their generation. So ಹೀಗಾಗಿ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್ ಆಗೋದ್ರಿಂದ ಏನಾಗುತ್ತೆ, ಅವರು ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನನ್ನು ಅವಾಯ್ಡ್ ಮಾಡ್ತಾರೆ. ಸೊ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್ ಆಗ್ತಾ ಆಗ್ತಾ ಸೊ ಆ ಒಂದು ಜನ್ರೇಷನ್ಸಲ್ಲಿ ವಿದಿನ್ ದರ್ ಜನ್ರೇಷನ್ಸ್ ದೇ ವಿಲ್ ಗೆಟ್ ಫ್ಯೂ what variations. So there are varied varieties. How nowadays we are getting varieties of mangoes, varieties of fruits, cereals, pulses, vegetables, in everything there are varieties. Okay, because so these all are meant for us only. Like we have like what is the food habits, cereals irbodu, pulses irbodu, vegetables na tintivi, ಪ್ರತಿಯೊಂದು ಕುಕಿಂಗಿಗೂ ನಾವು ಪ್ಲಾಂಟ್ ಸೋರ್ಸನ್ನು ಯೂಸ್ ಮಾಡ್ತೀವಿ, ಹೌದಾ? So just imagine if there is no varieties, where would we have survived? Okay, to survive we are the consumers, right? So producers should be very strong and the producer should be in varieties. Because of the varieties only we are consuming. Consumers are alive. Understood? Okay, so we'll study what are those outbreeding devices. There are many outbreeding devices, which is very important concept. 5 marks question will come, 1 marks question, all kind of questions they may ask. Okay, so we'll study one by one the outbreeding devices. So the first outbreeding devices are unisexuality, or it is also called as dicliny. What is that? Unisexuality and dicliny. Why? Because of bisexual there must be self-pollination. Bisexual flowers irodrinda enagutte, they will help to perform self-pollination. If they are not at all bisexual, if they are unisexual itself, then what happens? They must and should undergo cross-pollination. So hagagi plants will produce unisexual flowers. Okay, hence because of producing unisexual flowers, like example papaya, okay, date palm, coconut. So all these plants produce unisexual flowers, and what happens? They perform cross pollination. We know unisexual flowers idre enagutte, okay, from one complete a plant alli ಒಂದೇ ಕೈಂಡ್ ಆಫ್ ಫ್ಲವರ್ ಇತ್ತು ಅಂತ ತಿಳ್ಕೊಳ್ರಿ, ಲೈಕ್ ಸ್ಟಾಮಿನೇಟ್ ಫ್ಲವರ್ ಇತ್ತು ಅಂತ ತಿಳ್ಕೊಳ್ರಿ, ದರ್ ಇಸ್ ನೋ ಪಿಸ್ಟಿಲೇಟ್ ಇನ್ ದಟ್ ಪ್ಲಾಂಟ್. ಅವಾಗ ಏನಾಗುತ್ತೆ, ಮಸ್ಟ್ ಆ್ಯಂಡ್ ಶುಡ್ ದೇ ನೀಡ್ ಟು ಶುಡ್ ಟೇಕ್ ಹೆಲ್ಪ್ ಫ್ರಮ್ ದ ಅದರ್ ಪ್ಲಾಂಟ್ ಫ್ಲವರ್ಸ್. ಅದರ್ ಪ್ಲಾಂಟಲ್ಲಿ ಪಿಸ್ಟಿಲೇಟ್ ಇರ್ಬೋದು. ಸೊ ವಾಟ್ ಹ್ಯಾಪನ್ಸ್, ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್ ಪರ್ಫಾರ್ಮ್ ಮಾಡೋಕ್ಕಾಗಲ್ಲ, ಸ್ಟ್ರಿಕ್ಟ್ಲಿ ದೇ ನೀಡ್ ಟು ಪರ್ಫಾರ್ಮ್ cross pollination. This is one technique which is used by the plant to avoid self pollination. Okay, so the next device we call it as dichogamy. Dichogamy andre, so dichogamy means, so there will be difference in the maturation of stamen and pistil. So ಏನಾಗುತ್ತೆ, ಡೈಕೋಗ್ಯಾಮಿ ಅಂದ್ರೆ ಏನು ಹೇಳ್ರಿ? ಓಕೆ, ಇವಾಗ ನೋಡ್ರಿ, we have already studied. Okay, so not only normal anther and normal ovule is important for fertilization, that anther as well as ovule should get matured. ಹೌದಾ? ಮ್ಯಾಚೂರ್ ಆಗಬೇಕು. ಮ್ಯಾಚೂರ್ in the sense able to produce the gametes. Okay, so here also dichogamy ಏನಾಗುತ್ತೆ, so where there will be difference in the maturation time. Okay, maybe sometimes anthers quick ಆಗಿ ಮ್ಯಾಚೂರ್ ಆಗ್ಬೋದು compared to embryo sac or pistil, or first pistil will be get matured, then anther will be get matured. So higadaga there is no synchrony. As we studied in self-pollination, synchronization is very important. Yes, so here also if there is no synchrony itself, so they can't perform self-pollination. So based on the difference in the time of maturation of anther and stigma, so there are two kinds of dichogamy. Okay, so those are protandry and protogynous. Just remember androecium, gynoecium, protandry remembers or it indicates androecium. Protogynous, gynous indicates gynoecium. Okay, so protandry, what happens during protandry? The anther gets matured first compared to pistil. And what happens in protogynous? Our gynoecium get matured first, then androecium. This is what protandry and protogynous. These are the two different types of dichogamy. Okay, so next outbreeding device. So hercogamy, it is called as hercogamy, where what happens, there will be a physical barrier. Okay, physical or sometimes chemical barrier between pistil and stamen. So beautiful example for this, Calotropis. The barrier is pollinium. Okay, so ಏನಾಗುತ್ತೆ? So there will be a barrier, ಒಂದು ತಡೆಗೋಡೆ ಇದ್ದ ಹಾಗೆ. Just imagine this is a pistil and this is stamen. Between these two there will be a barrier like this. If there is a barrier, there is no transfer of pollen grain from this anther to the stigma. So there is a barrier, right? So they can avoid self pollination as much as possible. That is called as hercogamy. So next device is called as heterostele. So heterostele means there will be morphological difference. Okay, difference in the structure of stamen and pistil. So example, if this is the normal so this is our normal flower. Okay, this is our normal flower. In heterostele, what happens? Difference in the morphologically they are different. Who are different? Pistil and stamen. So hege andre, so here so this is pistil. So sometimes what happens, this anthers will be very short like this. These are one morphological difference. There is a height difference. Or sometimes this is the pistil and anthers will be like this. So because of different morphological arrangement of pistil and stamen, Okay, because of stamen and pistil different morphological arrangement, so they can avoid the self-pollination. This is what we call it as heterostele. Okay. So next barriers, or we can tell as devices, that is self-incompatibility. Self-incompatibility means by the pistil, like we know what happens, from the anther pollen grains are dispersed and they land on the surface of the stigma to perform the further post fertilization process or post pollination process, we call it as. So ಏನಾಗುತ್ತೆ, pollination ಆಗಿ anthers in the pollen grains ಎಲ್ಲಿ land ಆಗುತ್ತೆ, stigma surface ಮೇಲೆ. Okay, so now the stigma decides whether to allow pollen grain to form the pollen tube or not. Okay, that is what self assessment. The self incompatibility andre, this pistil or the stigma surface won't feel compatible for the pollen grain. So what happens, they won't allow the pollen grain itself to germinate on the surface of the stigma. So hige madodrinda ಏನಾಗುತ್ತೆ, self incompatibility formation ಆಗುತ್ತೆ. ಅದರಿಂದ stigma rejects the pollen grain. So like this they avoid self pollination. Understood? So these are the some outbreeding devices where they avoid the self pollination and encourage what, cross pollination to overcome inbreeding depression. Okay, yes. So this is what the outbreeding devices.